Greetings, curious minds. Today, we're embarking on a captivating exploration into the realm of pharmacognosy, a field where nature and science intertwine to unlock the secrets of medicinal plants and natural compounds. We, pharmacist moms, welcome you to this enlightening journey through the fascinating world of pharmacognosy. Before we dig into the wealth of knowledge awaiting us, don't forget to subscribe and hit the notification bell to stay updated on our journey through the pharmacy wonders. Now, let's peel back the layers of greenery and explore the captivating world of pharmacognosy. In this video, we will learn 1. Introduction to pharmacognosy. 2. History of pharmacognosy. 3. What is pharmacognosy? 4. Scope of pharmacognosy. 5. Relationship of pharmacognosy with other fields and professions, and 6. Future of pharmacognosy. Let's begin. Pharmacognosy factually means a knowledge of drugs or pharmaceuticals. It has been a part of the healing arts and sciences since mankind first began to treat illnesses. It has developed from ancient civilizations that used parts of plants and animals to formulate numerous potions to eliminate pain, control suffering, and counteract disease. It has risen from the mysterious incantations of voodoo tribes and has survived the unwritten secret recipes of medicine men. It has progressed from an era of experimentation to the present age of specific therapeutic agents. Nowadays, pharmacognosy is a highly specialized science, and it represents one of the major disciplines of pharmaceutic education. In order to gain proper perspective about a science that deals with plant and animal drugs and their constituents, let's survey past records and recognize those who have contributed to the subject matter that constituted the field of pharmacognosy in its beginning. By trial and error, primitive man must have acquired biologic knowledge that was useful in determining which plants and animals possessed food value and which were to be avoided because they were unpalatable, poisonous, or dangerous. His observations were handed down from one generation to another and were added to by his progeny. The healing powers of certain herbs, roots, and juices were undoubtedly discovered by accident, but once these attributes were learned, they were too important to be forgotten. The Babylonians made clay models of the human body, and early writings indicate that they were aware of the medicinal effects of a number of plants. It is a well-known fact that the ancient Egyptians were adept at embalming the dead, and that they possessed an understanding of the human anatomy, as well as a knowledge of the medicinal uses of many plants and animals, according to the Papyrus Ebers. This famous document, written in 1550 BC, was found in the tomb of a mummy, and is now preserved at the University of Leipzig. Dioscorides, a Greek physician who lived in the 1st century AD, wrote his De Materia Medica in 78 AD, in which he described about 600 plants that were known to have medicinal properties. Of these, a surprisingly large number are still important in modern medicine. Aloe, belladonna, colchicum, ergot, hyoscyamus, and opium are a few that were used then, in much the same manner as they are used today. Galen was a Greek pharmacist-physician who lived in Rome and who described the method of preparing formulas containing plant and animal drugs. He devoted considerable time to compiling this knowledge, which was distributed throughout 20 books. As a tribute to his accuracy in recording his observations, the term Galenical pharmacy was originated. From this humble beginning, medicine and pharmacy gradually emerged along separate paths. The physician diagnosed the ailment and prescribed the remedy, and the apothecary, or pharmacist specialized in the collection, preparation, and compounding of the substance. Thus, the term materia medica, meaning medicinal materials, was synonymous with the substances and products derived from natural sources, and was employed by the physicians of that era. The term pharmacognosy was introduced by C.A. Sadler, a medical student in Halle, also known as Sala, Germany, in 1815. This name is formed from two Greek words, pharmakon, drug, and gnosis, knowledge. The most comprehensive idea of the scope of pharmacognosy was presented by Flukiger, who stated that pharmacognosy is the simultaneous application of various scientific disciplines, with the object of acquiring knowledge of drugs, from every point of view. What is pharmacognosy? Pharmacognosy may be defined as an applied science that deals with the biologic, biochemical, and economic features of natural drugs and their constituents. It is a study of drugs that originate in the plant and animal kingdoms. What is the scope of pharmacognosy? Let's discuss. Possibly, as the lay public has heard little about the term pharmacognosy, there is an absence of recognition and, additionally, a lack of association of the term with the exact subject matter it signifies. However, an average person has intuitive curiosity who reads or hears of opium, morphine, foxglove, insulin, reserpine, thyroid, penicillin, blood plasma, polio vaccine, and even the much maligned castor oil. Numerous drugs used by the ancients are still employed, in much the same way, by today's medical practitioners. Even though the extraction, separation, isolation, and identification of the component constituents of plant and animal drugs that have happened in comparatively recent years, nonetheless the purpose for many of these medicinal substances employed these days matches closely the use for which they were proposed by our ancestors in the study of pharmacy and medicine. Modern aspects of the science include not only the crude drugs, but also their natural derivatives. Some examples include digitalis leaf and its isolated glycoside, digitoxin, rauwolfia root and its purified alkaloid, reserpine, and thyroid gland with its extracted hormone, thyroxine. In some cases, drug constituents have been partially replaced in commerce by synthetic compounds of identical chemical structure and therapeutic properties. Such natural and synthetic substances often can be distinguished by physical and chemical tests. For example, natural camphor is obtained from the camphor tree by steam distillation. It is dextrorotatory in its reaction to polarized light. In contrast, synthetic camphor may be manufactured by total synthesis from vinyl chloride and cyclopentadiene or by semisynthesis from pinene, derived from pine stumps, which is not entirely a synthetic process, but a chemical modification of a natural product. Synthetic camphor is racemic and can be differentiated easily from the natural form. Likewise, epinephrine, caffeine, codeine, ephedrine, menthol, penicillin, and other chemicals may also be obtained from either the natural source or by partial or total synthesis. They are considered a certain part of pharmacognosy. During the past few years, due to intense concern with all aspects of ecology, there has been a renewed interest in so-called natural foods and drugs. The accessibility of an enormously wide variety of these products, ranging from fenugreek tea to ginseng chewing gum, has encouraged the public to learn more about them. Subsequently, a massive literature on natural drugs is written by laymen to inform other laymen. Much of this literature is comparatively inaccurate and consists of beliefs and opinions substituted for facts. It is the pharmacist's responsibility to be aware of the existence of such pseudo-pharmacognostic writings, principally to be able to caution his patients concerning them and to correct any factual misinformation gained from reading them. In a broad sense, pharmacognosy holds a knowledge of the history, distribution, cultivation, collection, selection, preparation, commerce, identification, evaluation, preservation, and use of drugs and economic substances that affect the health of humans and other animals. These economic substances extend beyond the category of crude drugs and their derivatives to include a variety of commercial and medicinal products, often requiring complicated methods of preparation, allergens, allergenic extracts, antibiotics, immunizing biologies, flavoring agents, and condiments. In a constrained sense, the definition of pharmacognosy infers a specific knowledge of methods of identification and evaluation of drugs. How pharmacognosy is related with other fields and professions. Let's find out. As a part of the pharmaceutic curriculum, pharmacognosy forms an important link between pharmacology and medicinal chemistry on one hand, and between pharmaceutics and clinical pharmacy on the other. Pharmacology, like pharmacognosy, is an outgrowth of materia medica, the ancient science that dealt with all aspects of medicinal agents. Now, in this more specialized era, pharmacognosy deals primarily with information on the sources and constituents of natural drugs, and pharmacology is concerned with their actions and effects. The relationship of pharmacognosy to dispensing pharmacy and clinical pharmacy is clear when one considers the number of naturally derived drugs that are handled by the pharmacist in this age of drug specialties. Because of his knowledge of drug constituents, the pharmacist is able to predict not only the chemical and physical incompatibilities encountered in compounding, but also the therapeutic incompatibilities that the patient may encounter when utilizing a drug concomitantly with other prescribed or self-selected medications. Methods of procurement and preparation affect the price of drugs, thus, insofar as economics are concerned, pharmacognosy is closely associated with the phases of pharmacy administration that deal with prescription pricing. The pharmacist serves as an information source of all aspects of drugs to his colleagues in the medical, dental, and nursing professions, as he also provides information required for the safe and effective use of both prescription and over-the-counter, or OTC medications. These advisory roles are made possible by the vast background of the pharmacist, the drug expert, in fields such as pharmacognosy, pharmacology, medicinal chemistry, and pharmaceutics. Although the pharmacist is mainly concerned with those substances having application to public health, he realizes that many of these therapeutic aids are also utilized as beverages, as spices and condiments, in confectionaries, and as technical products. For example, coffee beans and tea leaves both yield caffeine, which has medicinal application, yet the original sources are mainstays in the diet of the American public. Likewise, winter or green oil and ginger are used pharmaceutically, but a much greater quantity of each is utilized by the soft drink industry. Similarly, mustard seed and clove have definite therapeutic application, but they are in more demand in the spice and condiment trade. In addition, cinnamon oil and peppermint oil are valuable carminatives, however, they enjoy an enviable reputation as popular flavoring agents in candies and chewing gums. Furthermore, certain industries depend on large supplies of rosin, turpentine, linseed oil, acacia, pectin, and numerous other natural products that have a relatively limited application in the field of pharmacy. Coming up are facts related to the future of pharmacognosy. In spite of outstanding record of past achievements, solid scientific research in the field of pharmacognosy is suffering today, and some have expressed concern for its future. With so much interest in plant drugs, why, then, has quality scientific research in the field fallen on such hard times? Let's explore. The reasons include problems in obtaining adequate patent protection, in the supply of raw materials, in acquiring appropriate personnel, in testing and evaluating plant extracts, in the attitudes of researchers, and the like. But they are not the basic reasons. The basic reason is that the programs have not been successful in yielding new, marketable medicines, with a single exception. For instance, the SmithKline and French company supported a screening program of alkaloid-containing plants for more than a decade without obtaining a single new product. 25 years of effort by researchers for the National Cancer Institute, who tested some 40,000 plants, did not identify a single new agent of general use in the treatment of human cancer. The only notable exception was the discovery of vincristine and vinblastine in the Madagascar periwinkle by researchers at Eli Lilly and Company. It is interesting to note that the Lilly researchers included the plant in their tests not because of its reputed anti-tumor properties, but because of its reputation as a hypoglycemic agent. Successful discovery and development of new plant drugs, in the strict regulatory climate that prevails in the United States, must be a broad interdisciplinary effort, involving experts in a number of fields, including plant taxonomy, ethnobotany, pharmacognosy, biochemistry, analytical chemistry, pharmacology, pharmaceutics, and medicine. Few organizations provide the opportunity to bring the expertise of such a large number of individuals to bear on a single problem. Educational institutions cannot do it easily because of the barriers erected along school and even departmental lines. Major pharmaceutical companies do have all the expertise required, and if used appropriately. Still, the lack of effective cooperation of researchers in a variety of fields accounts, in large measure, for the recent lack of success in the development of new plant drugs in this country. At the same time, research and development in the field continues to flourish in another nation that is quite similar to United States from the point of view of technologic advancement. That nation is the German Federal Republic, or West Germany. There, new plant drug preparations, so-called phytopharmaceuticals, and even new plant constituents are continually being introduced into the market by a relatively large number of manufacturers. A survey there showed that nearly 76% of the women interviewed drank herbal teas for their beneficial effects, and about 52% of them turned to herbal remedies for the initial treatment of minor illnesses. In addition, the regulatory climate regarding new drug introduction is much more favorable in Germany, and even some of the smaller companies have the resources to innovate in the plant drug field. This serves to stimulate competition and thereby encourages new product development. In Germany, standardization of plant drugs and of proven safety and efficacy is no particular problem with modern instruments and techniques. Safety of long-use natural products is generally assumed if no side effects have been reported. However, if evidence of toxicity is found, the drug is promptly withdrawn from the market. For example, Symphedom is no longer available in Germany because of its content of carcinogenic pyrrolizidine alkaloids. Proof of effectiveness of the German development programs is the area in which significant cost savings are afforded to the pharmaceutic industry. Substantial emphasis is placed on the reports of clinical experience supplied by general practitioners, and this is supplemented by evidence found in the literature and by data supplied by manufacturers. What is green wave? Let's discuss. One factor that may facilitate the growing influence of those concerned with the proper preservation and use of natural resources are sometimes referred to collectively as the green wave. Those consumers who now honestly believe that natural is better are not going to be satisfied with a "we don't know" answer when they ask if catnip really has a useful sedative effect, or if garlic actually reduces high blood pressure, or if ginseng definitely increases resistance to disease. Their ever-increasing political influence may force the government to act, probably in two ways. Due to perpetual shortage of funds, government may ease somewhat the unnecessarily rigid standards for marketing new drugs, particularly drugs from plants, and thus stimulate more producers to begin research and development of them. The healthy competition thus produced will provide the answers so long sought for some of the older drugs. It will also stimulate the search for new ones. There is certainly no end in sight of raw material awaiting investigation. Only about 5 to 10% of the 250,000 plant species have ever been studied either chemically or with respect to their physiologic activity. Still, very good ones remain undiscovered in this vast backlog. Improvement in screening procedures will be helpful in identifying new and useful drugs. One of the great uncertain blocks for those seeking drugs with new physiologic activities or in fractionating extracts to determine the identity of the active constituent has been the lack of simple bioassay procedures. Fairly elaborate screening techniques have existed, but they require the collaboration of skilled pharmacologists and are, in addition, expensive. Researchers have reported significant success with a simple bioassay utilizing brine shrimp, that is, Archmea salina. The viable eggs of this tiny creature are readily available in the dry state from pet shops, where they are stocked as food for tropical fish. After hatching in a brine solution, the shrimp are exposed to varying concentrations of the test material and an LC50, that is, median lethal concentration value, in microgram per ml, is calculated. It has been determined that the activities of a broad range of compounds are manifested as toxic to the shrimp. The method is rapid, reliable, inexpensive, and may be conveniently applied in-house by chemists, botanists, or others who lack the resources and the technical ability required to carry out standard bioassays. For just these reasons, it has been successfully utilized in several studies involving the screening of large numbers of plant species for compounds with useful biologic activities. Another technique of this sort that shows considerable promise for the future is the so-called potato disc assay. This involves observation of the inhibition of crown gall tumors induced on potato discs by Agrobacterium tumefaciens, by various plant extracts or constituents being screened in a pilot fashion for their anti-cancer potential. Results so far have been hopeful and have correlated well statistically with other much more elaborate and costly cell culture procedures, at least for certain kinds of anti-tumor activity. These methods must be seen as forerunners of even more simple and reliable procedures for determining bioactivity. In future, they will probably be remembered with the same degree of amazement and even disbelief we now express for the old-fashioned pregnancy test that involved injecting the patient's urine into a rabbit and then sacrificing the animal to examine the condition of its ovaries. The modern woman, suspecting pregnancy, simply dips a plastic strip bearing certain monoclonal antibodies into a urine specimen and looks for a color change. This kind of specific enzyme immunoassay, carried out at home by the patient herself, could scarcely have been envisioned years ago. Few years from now, our assay procedures for bioactive plant constituents will be similarly improved. Because of their complex nature, plants were once considered extremely difficult to fractionate for the purpose of isolation and identification of active constituents. The second half of previous century saw steady improvement in analytic techniques. Selective solvent extraction, as outlined by G. Dragendorf in the 1880s, gave way to adsorption chromatography, which in turn spawned ion exchange, paper partition, and thin layer techniques, before developing into high-pressure liquid chromatography, or HPLC, and gas chromatography. Coupled with mass spectrometry, this last methodology has been extremely useful. So-called multiple-stage, or tandem mass spectrometry, has now come upon the scene, permitting identification and quantitation of nanogram quantities of constituents in tissue samples, approximating 1 mg, without prior extraction or purification. Other powerful techniques, useful for structural determination of complex plant constituents include X-ray crystallography and nuclear magnetic resonance spectroscopy, in its several variations. It is evident that further modifications in all of these techniques, coupled perhaps with some unexpected new developments, will lead eventually to the ultimate black box analyzer. A few milligrams of plant tissue, placed in one side, will be converted into a printout on the other, listing all of the constituents of interest and their concentrations in the sample. This device, which even today is only a few steps away from reality, will certainly expedite the screening of plant materials for useful constituents. Coupled with improved and simplified bioassay techniques, it is bound to reveal numerous new and significant medicinal agents. One of the existing difficulties in producing plant drugs is securing adequate amounts of the proper raw material. Collection and quantity of properly identified, sometimes scarce, wild-growing plants is becoming increasingly difficult as civilization encroaches on once virgin territory, for example, the Amazon basin. This difficulty will be overcome in the years ahead by plant cell culture methodology. Production of large quantities of undifferentiated plant cells is already possible, although we now know that apparently homogeneous populations are, in fact, quite heterogeneous with respect to their ability to biosynthesize and accumulate desired secondary constituents. Selection procedures are thus necessary to obtain high-yielding subpopulations. Present limitations of plant cell culture techniques include slow growth, expensive media, and the tendency to store desired metabolites in the tissues rather than to excrete them into the media, where they are more easily recovered. Such production is now economically practicable only when the compounds produced are both costly and unique as plant products. Some compounds that may currently be produced in high yields in such cultures and approach qualifying under these guidelines include diasgenin from Diascorea deltoidea, serpentine from Catharanthus roseus, and ubiquinone 10 from Nicotiana tabacum. In the future, existing production techniques will be greatly improved as a result of new information regarding factors influencing the formation of secondary plant constituents. Research has already shown that certain pharmaceutically desirable secondary natural products are phytoalexins, that is, compounds synthesized only when the plant is subjected to physical, chemical, microbiologic, or fungal damage. Examples include gossypol in Gossypium arboreum and homoharingtonine, and related alkaloid esters in Cephalotaxis harringtonia. Increased yields of both of these compounds resulted from the addition of heat-denatured conidia of the wilt-producing fungus Verticillium dallii to cell suspension cultures. Yields of codeine and morphine in Papaver somniferum cell suspension culture were also greatly improved by similar methods. Substitute methods will be developed for plants unable to accumulate large cell masses in a relatively short time. In such cases, isolation and immobilization of either a key enzyme or the intact plant cells themselves seem feasible. Immobilized Papaver bracteatum cells have been shown capable of producing codeine from codianone at a bioreactor conversion of 20 to 30% and of maintaining the reaction over a period of 27 days. A desirable feature of the process is the fact that 88% of the codeine is excreted into the medium, where it can be easily recovered. Another approach to circumventing a slow growth rate in plant cell suspension cultures is to transfer the plant genes, which code for the enzymes catalyzing the desired biosynthetic reactions, into a bacterial or fungal cell. This could lower fermentation times to 24 to 48 hours, instead of the 6 to 8 weeks necessary for fermentation with a typical plant cell suspension. Many gene products are required to produce desired secondary metabolites. The technology to accomplish this is not yet available, but 1- or 2-step enzyme-catalyzed conversions should be possible now. More complicated reaction sequences will soon follow. In the future, successful commercial production of plant drugs by cell culture techniques will depend on the development of strategies that increase yields and shorten fermentation times. The manipulations of plant cells, plant genes, and plant enzymes, rather than of the plants themselves, should provide us with a host of useful medicinal agents. Diseases for which satisfactory cures still remain to be developed include viral diseases, such as herpes including genitalis, simplex, and zoster, AIDS, and certain cancers. Diseases of unknown etiology, including arthritis, some cancers, muscular dystrophy, and Parkinsonism. Self-inflicted diseases, such as alcoholism, drug dependency, obesity, smoking, and stress. Genetic diseases, ranging from cystic fibrosis and hemophilia to sickle cell disease. In addition, improved drugs need to be developed for the control of symptoms, such as pain. A non-addicting narcotic is, for example, urgently needed. Treatments are also required for conditions not readily positioned in any one of these categories, for instance, elevated cholesterol levels, hypertension, and even the general susceptibility to diseases of various kinds, both infectious and non-infectious. It is in this last area that some promising progress has already begun to occur with plant drugs, progress that evokes the promise of eventual breakthroughs of considerable importance. In recent years, the concept of immunotherapy, that is, the stimulation of the body to develop and improve its own defenses, has developed from a mere theoretical concept into a very promising field of drug therapy. Numerous compounds, affecting this advantageous process, by a variety of different mechanisms, have been cautiously identified in higher plants. Low molecular weight compounds comprise terpenoids, alkaloids, phenols or quinones, and lipids. Active compounds of high molecular weight are the glycoproteins, polysaccharides, nucleoproteins, and proteins. Constituents of both types have been identified in such well-known drug plants as Arnica montana, Baptisia tinctoria, Echinacea purpurea or E. angustifolia, Eupatorium cambinum or E. perfoliatum, and Thuja occidentalis. The sustained study of all aspects of these immunostimulants should prove a productive field of scientific endeavor in the future. Another part, in which plant principles are exhibiting considerable promise, is that of hepatoprotective drugs. Such agents are urgently needed, not only to protect the liver from infections, such as viral hepatitis, but also to help prevent damage from accidentally or purposely ingested toxins, ranging from amatoxins to ethyl alcohol. Silymarin, a mixture of flavonolignans, obtained from the seeds of the milk thistle, Silybum marianum, is currently marketed in Europe, and used for just these purposes, with considerable success. There are various traditional plant remedies that have long been used in Oriental medicine, for their anti-hepatotoxic properties. One of these that has gained popularity because it is currently being marketed here as an adaptogen, an agent that strengthens the resistance of the body to all forms of stress, is the fruit of Schisandra chinensis. Although the species has yielded more than 30 lignans, some of which were reported to suppress chemically induced liver damage in mice and rats, there is, as yet, very little evidence to substantiate the claim that it has useful hepatoprotective properties in human beings, or that it increases bodily resistance to disease or other forms of stress. What is needed with Schisandra and other similarly used plants, including the familiar turmeric, Curcuma longa, is additional scientific and clinical investigation. The so-called adaptogenic drugs, for example, ginseng or Panax species and Eleuthero or Acanthopanax senicosus, require detailed study to show if they do exert such a salubrious action. If so, we need to uncover the mechanism. How does it differ from immunostimulation? The exact identity of the active principles, and many similar questions that remain unanswered. If we are ultimately able to determine that the effects attributed to ginseng are not only real but significant, then this slow-growing root will become a prime candidate for the application of future cell culture methodology. Within this century, the accomplishments of science and technology will be so great that, when brought to bear upon the mysteries of nature that have long puzzled us, those mysteries will yield their secrets with incredible speediness. It will be a fascinating and eventful period. We will know not only the causes of disease, but the cures for most. The plant and animal kingdoms will continue to serve mankind in the 21st century, just as they have done since the dawn of history. Important new drugs of natural origin and new approaches of producing them will continue to be significant parts of that service. As we conclude our journey through the enchanting world of pharmacognosy, it's evident that this field is not just about studying plants, it's about understanding the delicate dance between nature and healing. From the rainforests to your backyard, every plant has a story to tell, and pharmacognosy allows us to listen, learn, and harness the medicinal treasures they hold. If this exploration has piqued your interest, we encourage you to continue discovering the wonders of pharmacognosy. Stay curious, stay connected, and join us in celebrating the profound synergy between nature and medicine. Thank you for joining us on this journey. If you found this video insightful, don't forget to give it a thumbs up, share it with your fellow explorers, and, of course, subscribe for more captivating content on the intersection of science and nature. Until next time, we, Equation by Pharmacist Moms, signing off from this adventure into the marvelous world of pharmacognosy. Take care, and keep exploring the beauty of the botanical realm.